afterwards.
Yeah.
Okay.
Hello, everyone. Welcome to this first session of the meeting after the preliminary speakers. It's each year at the annual meeting, the journals owned by the Society come together to create joint symposia. And the turn this year was for BJP and BJCP to collaborate. And we unanimously agreed, the editors-in-chief and the boards, the senior editor boards, that the advances in RNA-based therapies deserved the spotlight, hence our symposium. We've got an excellent lineup. I have to tell you, sadly, that one of our speakers is unable to attend due to unforeseen circumstances, Dr. Helen Lightfoot. But what this has meant is that our speakers can relax a little bit, and we'll be giving them a little bit of extra time in giving their talks, but also there's greater time for questions, and I hope that you will all take advantage of that. We'll be taking questions at the end of each of the talks. I should introduce myself. I'm Professor Amrita Al-Jamal. I am currently the editor-in-chief of the British Journal of Pharmacology, although I won't be for much longer. My tenure ends at the end of this year. And I'm here with my colleague, who I shall let introduce himself.
Oh, okay, thank you. So my name is Serge Cremers. I'm a professor at Columbia University in New York City, and I'm the editor-in-chief of the British Journal of Clinical Pharmacology. And unless they get rid of me this year, I will be for another few years. So thank you. This is a very exciting session with where we literally span preclinical to clinical and across various disease areas, as well as even areas within pharmacology. So very much looking forward to this. And now let Amrita introduce the first speaker.
At the end of the talks, you can ask questions. There are microphones. I can't actually see now, but I think they're by the numbers somewhere in the aisles. If you could go to those microphones, those standing microphones to ask your speakers, we'll then point to you and you can ask your question. We do also have people attending virtually, and we have some BPS staff who are going to be helping us to address their questions. So those of you who are watching the live stream through the app, you can ask your questions. Please make your questions as the talk's going, and then the staff will pick them up and we will attempt to get them answered for you. Okay, so I'll introduce the first speaker. I'm not sure that I really need to, actually. It's Professor David Webb. I'm sure many of you know him very, very well. He is a physician and a clinical pharmacologist at the University of Edinburgh. He directs the Hypertension Excellence Centre at Edinburgh University, and also created the Centre for Cardiovascular Science at Edinburgh. He is also a theme lead in the BHF Centre of Research Excellence. His research, of course, has focused on hypertension research. He's made some amazing discoveries, not least the very recent work on paracetamol, which has been very exciting. He's a past president of the BPS, and he's also president of the World Congress of Pharmacology, which is going to be in Glasgow, July 2023. He's a fellow of the Royal Society of Edinburgh, Academy of Medical Sciences, and in 2020, he was awarded a CBE in the Queen's Birthday Honours list. And he's going to talk to us about hypertension and silencing RNA. David.
I wish my mother could have heard that. Well, thank you for the invitation, and it's really nice to be back at what is at least partly a face-to-face meeting, so I'm glad there are people in the audience today. And as you've heard, I'm going to talk about an siRNA for hypertension. I am a clinician, and I will look largely at clinical aspects, but I will give an introduction to siRNA at the beginning, and this work has been done with colleagues in Edinburgh, and this talk's been put together with my colleagues Dr. Mel Addison and Professor Priyanka Ranasinghe. So let's start. So declarations of interest. I was the principal investigator in Edinburgh for the phase 1 studies with xylobesaran, which was known as ALN-AGT. and that comes from Al-Jamal Pharmaceuticals. And I have been a scientific advisor for a number of companies. They've all been linked to hypertension projects, and they are all with departmental or non-personal funding. So I haven't received funding from these collaborations. So what I'd like to do is give a brief introduction to hypertension, explain why it's important, talk about its current management and unmet needs, particularly in relation to siRNA, give an introduction to siRNA therapeutics, and then focus on zilebesiran, which is a drug that knocks down angiotensinogen. I'll summarize the clinical evidence on efficacy and safety, and I'll consider the clinical potential, some of the challenges that might exist, and where we're going to go next, and I'll then summarize with a few key messages. So, I probably don't need to tell you this, but hypertension is the global leading cardiovascular risk. If you look at the figure from the on the left from the risk factor collaborators on global burden of disease, this is from 2020. It's the most important risk factor, and also because it's above that horizontal line there, it's increasing rather than decreasing. And it currently affects probably more than 1 in 3 adults, and it kills 10 million people annually. So it's a significant problem. And if we look at the costs, then there are substantial costs, largely through stroke and coronary disease, also through vascular dementia and chronic kidney disease, as well as aortic dissection, heart failure, and a number of other conditions, including atrial fibrillation. But you can see that 3/4 of the financial burden is from stroke and coronary heart disease, and is a major pressure on the health service in the UK, 2 billion in England. So, why do we need a new drug for hypertension? We certainly have lots. After trying lifestyle modifications, which are not always successful, we use oral medications, usually once daily, and we now have 68 different medications for hypertension in 15 different classes. They're generally well tolerated and mostly reduce blood pressure by about 5 to 10 millimeters of mercury. And they've been shown to work and reduce deaths and disability through very large clinical trials. Some of the largest trials have been in hypertension. However, the last class of drugs that were launched were licensed over 15 years ago, and those were the direct renin inhibitors. And it has to be said, they haven't made a huge impact on the way we treat hypertension. So again, why do we need a new drug? And that's because we have huge challenges when we treat hypertension. First of all, and I'm not going to deal with this in this talk, but almost 50% of people with hypertension are undiagnosed. And that's because we just don't measure blood pressure in the population in order to protect them from high blood pressure. The second area in which we do detect hypertension is poor treatment. So only 20% of those who are detected have controlled blood pressure. And that's partly because some of them don't get treated, and partly because of what's called therapeutic inertia, which is when the clinician involved detects hypertension, starts a medicine, and doesn't go back to see how well it worked. They don't increase the dose. They don't start a second medication if that's required. So that's one problem. And then the second problem is patients don't take their tablets. And that's a really big problem. Almost half of the people with hypertension who start treatment are not taking all of it a year later. It's partly because I think it's not seen as important by patients. It's asymptomatic. It's a lifelong condition to take tablets for, and a lot of patients only start taking them when they unfortunately have a stroke or heart attack. Sometimes it's because patients are on complex treatment regimens with multiple tablets. They find that hard to manage. And of course, sometimes people get get side effects and don't think to discuss it with their doctor. They just stop taking the tablets. So non-adherence is a significant problem where these new siRNA agents may be important. So what are short interfering RNAs, or siRNAs? Well, they're short double-stranded RNA, with a sense and antisense strand. They're fairly short, 20 to 24 base pairs, molecular weight of 13 to 14 kilodaltons. So they're generally smaller than antisense oligonucleotides. And they have the potential to knock out proteins or knock down proteins that wouldn't be easily druggable through conventional means. So this is the mechanism of action. So if you follow the numbers here, so for a drug that's in the circulation, an siRNA in the circulation, it has to get into cells first of all. So it has to cross the cell membrane, usually by being incorporated into endosomes. From endosomes, stage 2, they have to escape from the endosome, and they have to be lucky enough, before they're destroyed, degraded, to get bound into the RISC complex. That's the RNA-induced silencing complex. Once that happens, then one of the strands, the non-guide strand, is lost, and the single strand that's left binds its complementary RNA, which is the target for the protein of interest. And then once the target mRNA has been incorporated into RISC, it gets degraded, and the RISC complex can be recycled to pick up another target mRNA molecule. So there have been multiple challenges. So it's hard to get siRNAs into cells. There's fairly low levels of endosomal escape. When these agents are in the circulation, they're easily and rapidly renally cleared. They're easily degraded by nucleases. And then, as well as the difficulty of getting it to the right place, there's side effects that are potentially a problem, like immune activation and off-target effects on other RNA. So the solutions I won't go into in detail, but there have been some pretty clever chemical modifications to improve the way these RNA perform, and then delivery strategies which have involved either encapsulation, for instance, in lipid nanoparticles, or ligand conjugation. And the particular one I want to talk about is GalNAc, N-acetylgalactosamine, which has been transformative. That's made a huge difference. So, why is GalNAc wonderful, and why is it wonderful for the particular protein I'm interested in, angiotensinogen? Well, GalNAc binds to the asialoglycoprotein receptor on hepatic cells, so it goes straight to the liver. These receptors are only expressed on the liver, and they're in high abundance and rapidly recycle. So it's easy to get a GalNAc compound into the cell. They then sit in the endosomes. The biochemical changes that have been made mean that they sit there and can reside for days, weeks, sometimes months. And then when they do escape, they go through the normal process, and RISC recycling, that stage 7, means that they can keep on knocking down angiotensinogen mRNA. So they can have quite a long action. And that's really the attraction, I think, the safety and the long action. So I'll just give you a very brief timeline. Fire and Mello discovered RNA interference in 1998. Very soon after that, it was shown in mammalian cells, and then in mice. And then the first clinical study, not necessarily a brilliant study, but was local administration into tumours back in 2004, meant that they won the Nobel Prize for their discovery. And you'll see at the top in the middle, Nair and colleagues demonstrated the GalNAc technology. And following that was 2014, we've had a flurry of drugs, not only developed, but licensed. First of all, patisiran, givosiran, and lumasiran, all for rare liver diseases. And then, more excitingly, from the point of view of hypertension, very recently in 2001, inclisiran, which knocks down PCSK9, was licensed for hypercholesterolemia. And that drug is obviously for a common chronic condition. And it's not only been licensed in the UK, US and Europe, but it's also been approved in appropriate patients for use by NICE in the UK. So that gives you some promise for the new development in hypertension. Why angiotensinogen? Well, it does target a system we've already targeted several times before. So angiotensinogen is the sole precursor of all angiotensin peptides, and of course angiotensin 2 acts on the AT1 receptor, and it causes effects on hormones, the heart, blood vessels, the sympathetic nervous system. All of those effects increase blood pressure. So it's well proven from the use of ACE inhibitors and angiotensin receptor blockers to be a good target for development. And zilebesiran is the first in class that attacks this particular system. As I said, it's a chemically modified GalNAc conjugated siRNA. It just attacks liver angiotensinogen, and it's currently now in phase 2 clinical development. And I'm going to show you the phase 1 data, the interim data that are in the public domain, the papers at the moment under review for publication. So this is the phase 1 first in human study, a classical ascending dose study with overlapping cohorts. It was a multi-centre with all centres in the UK, in Edinburgh, London and Manchester. It was a double-blind randomised trial with a 2-to-1 randomisation. So for each of the cohorts, there were 8 patients who received zilebesiran and 4 who received placebo. These were hypertensive patients. They could be on medication or not, but if they were on medication, they had to wash out for 2 weeks, which is the WO at the beginning of the treatment period. And if you look at the bottom right of that figure, you'll see they had a single dose on day 1. The study was supposed to end on day 84 after that single dose, but it's been substantially extended for reasons that will become obvious in the next few slides. So, if we look at the effects on angiotensinogen, placebo is the grey at the top, no real effect. And then you can see that for all of the active doses, there's a slow onset reduction in plasma levels of angiotensinogen. You can see that it's a dose-dependent effect, with the highest dose producing over 90% reduction out to 12 weeks, and actually the top 3 doses produced over 90% inhibition out to 12 weeks, and for the higher doses, and for these data, the 800 milligram dose hadn't gone out to 48 weeks, but I think we can safely assume that it would have had at least a great effect as the other 2 doses down, and so very substantial 80% inhibition even out to 48 weeks. If we look at blood pressure, ambulatory blood pressure was measured in this study. These are the 8-week data, and the light blue is diastolic pressure, and the dark blue is systolic blood pressure. We tend to focus on systolic blood pressure. It's probably a better predictor of outcome. And if you look at the far left, that's the 8-week data for placebo, really no effect on 24-hour ambulatory blood pressure. But for both diastolic and systolic pressure, you can see that there is a roughly dose-dependent effect, with the largest dose, 800 milligrams, which at this time had only gone out to 8 weeks, having an effect of over 15 millimeters of mercury on blood pressure. So that's a pretty big effect, and I can tell you that is probably an underestimate of the total effect when the trial finished. So why would it be different? Why would it have a bigger effect than an ACE inhibitor or an ARB? And there are lots of reasons why that might be, but one of these is something that's well known to those who work in heart failure and hypertension, which is called RAS escape, renin-angiotensin system escape. So if you block the angiotensin receptor, or if you block ACE, then angiotensin 2 levels go down, or angiotensin 2 effects on the AT1 receptor reduce, and that stimulates renin secretion from the kidney. Very large amounts of renin are produced, and then the precursor peptides are produced. So there's essentially competition at ACE or competition at the angiotensin receptor, and that, to some extent, probably attenuates the full effect of giving a renin-angiotensin blocker of the classical sort. In the case of angiotensin siRNA, the knockdown is so substantial that there isn't really any peptide, although renin levels will go up to very high levels and have been shown to do so in animals, there's really not much of a substrate to work on. Hence, that may be an explanation for why the effect's so large. What about safety? The drug looks safe. This is only phase 1. It's only 84 subjects, 56 of whom had xylobesaran, but most of the adverse effects were mild to moderate in severity, and they resolved without any need for intervention. There were no deaths, there were no treatment-related serious adverse events, none leading to study withdrawal, no patient required intervention for low blood pressure, and there were no clinically significant elevations in either ALT, which would mark liver damage, creatinine, which might mark renal impairment, or potassium, which might be a biochemical effect of knocking this system down. 5 patients had injection site reactions, but they were mild and transient, and those 5 were all patients who had received xylobesaran. So what's the advantage? Why is this potentially exciting? Well, the slow onset effect avoids a problem that has occurred with ACE inhibitors, which is first dose hypotension. So it takes days or weeks before the blood pressure comes down. And then that long-term effect with a single subcutaneous dose could be a solution to adherence problems that people have with daily dosing. We've seen a nearly complete knockdown of angiotensinogen. We don't know, but it's possible the effects with a second dose and third dose will even further accumulate the reduction in angiotensinogen and blood pressure. That waits to be shown. There's the potential to avoid RAS escape, and these drugs have been so far well tolerated. There are some questions. Will the clinical effect of repeat dosing be robust in phase 2? And there's a study, Cardia-1, that's going to address that problem with repeated dosing. There's a concern about antibody-mediated loss of efficacy, but that wasn't seen as an issue in the phase 1 study and hasn't been seen to any extent with a similarly engineered molecule, which is inclisiran for hypercholesterolemia. So I think that's less likely to be a problem. We need to know whether it works in combination with standard ACE inhibitor, ARB, calcium antagonist, and diuretic treatment, and that's the subject of the other phase 2 study, Cardia-2. In the longer term, we need to know whether these drugs will be useful in treatment-resistant hypertension, because these are the highest risk patients, and there is quite a high degree of poor adherence in this group. And of course, the most important question, which will only be resolved by very large clinical trials, is whether it improves long-term outcomes, cardiovascular events, renal protection, and reduction in mortality. In terms of safety, what we don't know is how this drug will perform in high-risk patients, so people with diabetes, heart failure, and the like. And of course, in elderly patients, the upper limit age for the phase 1 studies was 65. There is a concern, I share a concern, that there may be patients in which you wish to rapidly reverse the blood pressure lowering with this drug, such as after hypovolemia, hemorrhage. I'll talk about that again in a moment. And whether it might cause problems like acute kidney injury when it's combined with other RAS blockers. That's going to be studied in Cardia-2, but currently EMA advice is not to combine any 2 of ACE inhibitors, ARBs, and direct renin inhibitors. So that, I think, is another issue that needs to be addressed. So briefly on reversal, it's obviously in an emergency, if somebody has a substantial hemorrhage or becomes septic, that blood pressure lowering needs to be reversed to prevent AKI and organ damage. And the ways that have been suggested to do this are, first of all, oral or IV sodium chloride. And in the phase 1 study, Part B looked at high-salt diet. Vasopressors, noradrenaline, and angiotensin II both work to reverse hypotension preclinically in rats. Again, fludrocortisone will reverse the hypotension with xylobesaran in rats. And finally, somewhat elegant, but whether this would ever be used, I'm not sure, but there was a paper in Nature Biotechnology a couple of years ago showing that oligonucleotides complementary to the siRNA could reverse the effects in mice. So that's another potential approach. So there are other parts of the phase 1 study. There's a part in obesity which I haven't shown because there's some suggestion that angiotensinogen is associated, high levels are associated with obesity. But there are this part B, which looks at low salt, high salt diet, and high salt diet can reverse the blood pressure lowering of xylobesaran. And there's another study in which xylobesaran was given with irbesartan to address that question of combination. And in that study, additional hypotension was seen by adding irbesartan, but there was no evidence of renal impairment or increase in serum potassium. So in a phase 1 study, that appeared safe. So to summarize, the key messages here are hypertension remains a global health priority, and adherence is a key unmet need. Xylobesaran targets angiotensinogen, and it's the sole source of angiotensin peptides at the root of the renin-angiotensin system. It seems to be very effective in lowering blood pressure and acceptably safe in phase 1 studies to move to phase 2. Hypotension and hypovolemia are potential issues where options have been and are being tested, and efficacy and safety will be further addressed in the phase 2 studies. And ultimately, this may provide a very neat solution to the problems of adherence in patients with hypertension in clinical practice. I shouldn't leave without mentioning World Pharma next year, where Amrita and I have put together the scientific program, and we're talking about this afternoon if you have time to come and speak to us and ask questions about the meeting. And finally, I'd like to thank you for listening. And this is a picture of Edinburgh, a lovely city to visit when you come to World Pharma in Glasgow. Thank you.
You guys must be really happy. That was an amazing blood pressure lowering response. I don't know whether I missed it, David. All of the patients that were recruited in this trial, they were asked to come off their meds. So these are individuals who were taking blood pressure medicines for their hypertension. They came off them, and then after they'd had their washout, then they entered the study.
Yeah, so they had to have a blood pressure above 135 and below 165 at entry. They either weren't on medicines, so we had some who were newly diagnosed, probably half to a third were newly diagnosed, identified by ambulatory blood pressure monitoring, and the other half were on one treatment and were willing to come off treatment to join the trial. And then they had to have a washout and then a 24-hour ambulatory blood pressure that was above 130 systolic 24-hour blood pressure to get the subcutaneous injection.
Right, okay. So do you think that as a treatment, I mean, because patients respond to different blood pressure meds and you guys have lots of drugs that you can choose from, do you think that actually this might be an approach that could be used in a sort of, I guess maybe a semi-personalised approach if there are individuals who respond to drugs targeting the angiotensin pathways, that this might be useful, particularly in those individuals? Or is it that you're actually thinking that even if there are individuals who haven't had any blood pressure lowering response to ARBs or ACE inhibitors, that they would benefit from targeting angiotensinogen in this way?
I think at the moment, I think it needs to be tested whether it would work for all comers. It's a big effect. That it is actually more than 15 when you look at the final data. And it's people with quite low blood pressures at entry. I mean, it's only sort of 135 average. So that the drop you get with a blood pressure drug is generally proportional to the initial blood pressure. So more than 15 millimeters is really quite substantial. It's quite impressive. So I think this probably is worth looking at widely, assuming that it maintains its safety in phase 2. We do have a really good biomarker, actually, in hypertension that nobody uses, which is renin. And there's so many good studies that have shown that people with high renin are likely to respond to blockers of the renin-angiotensin system, and people with lower renins are less likely to respond. But we never seem to have taken that anywhere, which is disappointing. But we do something like it. We say that if you're old, or if you're Afro-Caribbean, which are groups that tend to have low renins, then you don't get a renin-angiotensin blocker. And if you're young, or if you're Caucasian, or if you're white, you start with a renin-angiotensin system blocker. So we do a surrogate version of that, but many people, including Maurice Brown, one of my colleagues, has argued strongly that we should actually just measure renin.
Thank you. I have more questions, but there are people in the audience who want to ask.
Oh, Martin Michel, Mainz, Germany. David, great talk.
Thank you.
I have one minor technical and two more philosophical questions, since Amrita said we have plenty of time today. Technical question. Those who were on hypertension meds before they entered the trial, how does the end of, and you have probably 30, 40 of them, how does the end of study blood pressure compare to the last blood pressure they had under the classic oral meds?
I can't answer that question.
Okay.
I can't answer it for two reasons. One, I actually don't know the answer, and if I did, I probably shouldn't tell you. But yeah.
That's fine. Lose lip sync ships. The other two questions I have is, when you talked about the RAS escape phenomena, there have been, over time, several people arguing that the increase in angiotensin with blocked AT1 receptors may even be a good thing by stimulating AT2s, which in several ways do that, which this approach would not have. So if you care to speculate about that. And the other was, you outlined a strategy.
Shall I just deal with that one first?
Sure, please.
I've got such a short attention span. So I think that's a really interesting concept. And it is possible that the AT2 receptor is important, but many people have tried to drug that and nothing's got past phase 1, which makes me think in phase 1 it does nothing. So I think it's a theoretical issue. The whole of that pathway is, it's shown to be present, but whether it has any meaningful value, I don't know.
Yeah.
And maybe this study will test some of that question.
Okay. The second question is, you outlined your strategy because on the long duration of action, if hypotension occurs, and the long duration of action, of course, is a blessing, but not only a blessing, but also occurs if you have anything serious happening other than low blood pressure. It may be difficult to get out of that within your 48 weeks. And how are you looking into that? Of course, you don't have seen anything serious yet, but do you have a strategy for that?
Yeah, I mean, that's not for me to answer. That would be for the company to answer. But I do have thoughts about that. You're absolutely right. I see that as the potential problem here. So I imagine that in most cases, the hypotension is not going to be such that you want it gone forever. You might want it gone for a relatively short period. And so that's why there's been an exploration of acute treatment. So if somebody gets a bit hypotensive, then one could up salt intake. If somebody becomes very hypotensive or has something else happen to them, then you've got intravenous approaches that are possible, and you've got fludrocortisone. I think if there is the really long-term issue, then maybe the idea of using a reversier, one of these small interfering RNAs, to reverse the effect of the original siRNA is possible, but I'm always not very attracted by the idea of using one drug to reverse the effects of another.
Thank you, Professor Al-Jamal.
Very, very interesting, David. So I go back to targeting the hepatocyte to silence the angiotensinogen. So is the liver the only place where this protein is synthesized?
That's a really good question. And I don't think we absolutely know the answer. A group of colleagues in the Netherlands who have a really long history of working with the renin-angiotensin system had originally talked about local renin-angiotensin systems in the liver, in the kidney, and elsewhere. So if there was an independent renin-angiotensin system in the kidney, and we are definitely seeing here a knockdown only of hepatic angiotensinogen, then it's possible that would be why, and in the preclinical studies, I probably should have said, in the preclinical studies, people have tried to cause acute kidney injury and haven't found it possible to do so in mice and rats. And it's been argued that's because the renal renin-angiotensin system is still functioning. The latest research suggests that may not be so, but I think that's an unresolved question, and we need sort of more definitive studies to help us understand that. But that's a really key question, because if that system was still in operation, that might explain why the kidney can be protected while angiotensinogen levels and blood pressure is very low.
Mm-hmm. And then it's about the reversibility, which could be, a solution to the problem, because with siRNA the effect is transient, especially when you're giving it in a non-viral way. And how long would it take, you think, after stopping the treatment for the enzyme to protein to be regenerated?
So with the 800 milligram dose, the single dose, probably can say this, I mean, it goes on for, it's still more than 70% knockdown at 48 weeks. So it's very substantial and quite long lasting. And the phase 2 studies, interestingly, are with a slightly lower dose, 600 milligrams. So I think that's probably been given some thought about how long. I think the aim would be, as for inclisiran, to be able to give a drug twice a year to get a continuous effect. And I think one of the reasons it has such a big effect is because, one, it's always there. You take it once and it's always, and however good people feel they are, even in clinical trials, at complying with the medicine, I think they probably miss some tablets, or some patients miss some tablets. Whereas with this, if you get the subcutaneous injection, you have the full effect of treatment. And the other thing is that whilst we give drugs once daily for hypertension, we know some of them don't last 24 hours, whereas this clearly does last 24 hours, and that's shown by the ABPMs at the beginning and at the end. The profile just moves straight down, more day and night pressures move down. So this is a very effective, long-standing approach.
So the frequency you expect it to be quite sparse?
6 months, I think, is what's aimed for.
Okay.
Yeah.
Okay, and do you know anything about receptor sensitization? For example, what happens if you want to give the angiotensin to agonist to the patient? And
Yeah. So in the rats, not surprisingly, they're slightly more sensitive to angiotensin than they would have been before they got the siRNA.
Okay, thank you very much.
Thank you. We should probably end, but just one quick question. In the placebo arm, I saw angiotensinogen go up. Is that right?
A bit, yes.
And there was no change in blood pressure at all in those individuals. Was that a consequence of do you think, the washout of the drugs that did that?
I don't know.
No.
I don't know. It's a modest change compared to the changes with active treatment. Things do tend to float upwards or downwards anyway with placebo.
And the knockdown, the 70% knockdown, was that still, was the blood pressure lowering still retained at the same level with the 70%?
Can't tell you that.
Okay. Thank you very much, David. That was fascinating. Thank you. We'll move on to our next speaker. Sorry.
All right. Thank you. So we'll move on to the next speaker, Professor Webb reduced it to one slide on formulation solutions, which is obviously has much more to it. And therefore, one of the specialists on this area on formulation in general is the next speaker, Professor Khuloud Al-Jamal from King's College. She is the chair in drug delivery and nanomedicine and head of medicines development at the Institute of Pharmaceutical Sciences at King's College in London. She has developed an extensive experience in designing and developing novel nanoscale delivery systems, including dendrimers, polymers, liposomes, exosomes, what have you. Her current work involves preclinical translation of nanomedicines with special interest in brain diseases and cancer. She received a number of awards and prizes as a real rising star. And so we're very happy to have her here talking about formulations. I can't even read it without glasses, so if you could take it from here.
Thank you very much, Serge. Thanks to the audience. I probably am wondering why I'm here, but I'll try to make my presence relevant to the Pharmacological Society, being a pharmacist and passionate about pharmacology. So I'll try to make the title slightly simpler, so ignore that for now. So that's next. Okay, so I'll give you some background about nanomedicine and why do we need delivery. So very relevant to the talk of David. We make many therapeutics that could be interesting in culture, in animals, but sometimes you drop them because of the side effects. And that's because these drugs do not make it to the site of action where you really want them to act, and you don't want them to go somewhere where they induce toxicity. And this is what we refer to as having a wide therapeutic index. So the range between toxicity and safety is quite high. So what we do with drug delivery, we try to have that precision in delivery to the site of action. So ultimately, we want to have the drug at highest concentration at the site of action, minimal somewhere else, and then what you then can do is push the dose without having the obvious toxicity, which could potentially translate into efficacy of the drug. So we work with small molecule drugs, nucleic acids, proteins, because now drugs are no longer small molecules. So what I'm going to demonstrate to you some of the case studies where we try drug combinations to improve the therapeutic outcome. So I'm more interested in cancer immunotherapy. So as Serge said, we are interested in treating cancer using novel modalities. So as you probably know, there are different modalities to treat cancer. There is chemotherapy, radiotherapy, immunotherapy, and one of the very recent approaches is to use immunotherapy that is activating our immune system to tackle cancer. And again, there are different submodalities to do so. So in this particular example, you may know a class of drugs called biphosphonates, and these are highly bone-binding drugs that are used for osteoporosis. But there has been studies to show that if you give biphosphonate to patients, you are able to make their gamma delta T cells, which are immune cells that can tackle cancer, proliferate. So what happens if you are able to deliver these biphosphonates to solid tumors? You probably are going to proliferate the gamma delta T cells and activate the immune system. But biphosphonates, we all know that they are rapidly excreted through the kidney, and they have very low affinity to solid tumors. So I was approached by a clinician who works with gamma delta T cells, and he said, are you able to redirect biphosphonates so they are highly accumulated in solid tumors? So what we've done, this work was back in 2012. We packaged these biphosphonates into what we call liposomes or lipid bilayer carriers. So if you look at this, you probably would think of the cell membrane, where you have phospholipids that are arranged into vesicular structures, but these are really tiny particles that are approximately 100 nanometers in size. And we use what we call passive targeting approach to reach the tumor. And what we refer to by passive targeting, you're relying on the physiology of the tumor, which has leaky vasculature. So these particulates would accumulate in the tumor, and because the tumor has poor lymphatic drainage, they would be retained there. This is what we call the EPR effect, the enhanced permeation and retention effect. So with passive targeting, we are able to package these biphosphonates and make them go to the tumor mass, and we have shown several reports that if you combine this treatment of liposomal alendronate with gamma delta T cells, you are likely to have a synergistic activity in solid lung tumors. So this was the beginning of using drugs to cause immunotherapy. And then we tried to combine this into different modalities. So how about combining chemo drugs with immunotherapeutic approaches? So Adam, who's now a scientist at AstraZeneca, has worked on a project where we wanted to deliver a drug, in this case doxorubicin. Doxorubicin is an anti-cancer drug, but recently it has been known to also have an immunogenic cell death activity. And what do we mean by immunogenic cell death activity? Is the cancer cell is dying in a way that is characteristic, apoptotic pathway that then would make our body recognize these tumor antigens and potentially vaccinate us against tumor recurrence. So doxorubicin is one of the few drugs that has this property, and we would like to combine it with an immune adjuvant, a nucleic acid-based immune adjuvant that can activate our immune system. But we want to co-deliver the anti-cancer drug together with the immune adjuvant to the same cells. So how can we do that? So here is the solid tumor, and what you can imagine, you're giving one injection that contains the doxorubicin solution, the drug, but also a service-reactive immune adjuvant, in this case, the CpG. And you're administering into the tumor mass. The drug will be taken by these cells. The CpG will functionalize the cell membrane, and we have used two approaches. One is based on maleimide chemistry, and one is used on cholesterol insertion. So these cells start dying, and they shed their apoptotic bodies that are also functionalized with the CpG immune adjuvant. And the apoptotic bodies are draining into the lymph node, where they basically present the fragments of the cancer cells to the immune cells, and they produce that specific anti-tumor response. So what we found was if you're treating those tumors implanted with a colon cancer model, and you look at their tumor growth, the non-treated tumor exhibited tumor growth that forces you to kill these mice in 3 weeks. But if you are delivering the individual treatments or the combination of the chemotherapy with the immune adjuvant, that you have significantly much slower growth of that tumor. So we believe that if you combine chemotherapy with immunotherapy, this is a good approach. But what we had to do here is really administer the treatment directly into the solid tumor, which is not always feasible. This is feasible with tumors where you can see them, such as melanoma, head and neck cancer, and breast cancer, and so on. So we assess the immune response, and what we can see that if you quantify the number of the CD8 cells in the tumor that receive the combinatory treatment, there are higher number of these CD4, CD8 cells into the tumor, and that probably correlated well with a reduction in tumor growth. So we wanted then to have a more versatile approach where you can administer these lipid nanoparticles intravenously. You want to deliver the doxorubicin systemically and rely on the EPR effect to reach the solid tumor. And that coincided very nicely with the approval of the first lipid nanoparticle formulation in 2018, and then we were hit with COVID, and we realized that lipid nanoparticles and nucleic acids are no far to realization as current therapies. So we were already working on encapsulating an siRNA with doxorubicin. So we kept the immunogenic cell death drug in the formulation, but we wanted to add a new siRNA to complement that activity. And what we really aimed to do here is silence a receptor on cancer cells called CD47. So cancer cells express a receptor called CD47, and CD47 is a do not eat me marker, so cancer cells hide themselves from the immune cells and macrophages, so the macrophages are unable to engulf them. So what about delivering a lipid nanoparticles that contains that siRNA, but at the same time deliver a drug that improves the expression of keraticolin receptor, which induces immunogenic cell death. So we designed these lipid nanoparticles that contain both the drug and the nucleic acid. So what you have to do here is design your lipid nanoparticles where it contains 4 lipid mixture. One of them is a charged lipid, or what we call ionizable lipid. So this ionizable lipid will be protonated at acidic pH, which is the pH we use to formulate these nanoparticles, and then after encapsulation, you bring the pH up so that they are neutral in charge and then safe to administer to the bloodstream. So what we think should happen is that you silence the CD47 and you overexpress the keraticolin protein, which activate the macrophages and help to clear these cancer cells from the body. So as a proof of concept, we designed this preclinical study. We gave the treatment to the mice with the individual treatments or the combination, and we monitored tumor volume as well as survival and other immune markers. So what's interesting here is the treatment that had the combination had significantly much smaller volume, but also these mice were freed from the tumor. So this is the number of tumor-free mice, and you can see the combination treatment, almost 60% of the mice cleared that tumor without any significant changes in the body weight. And you can see here, for example, the spleen weight, which is normally enlarged in this type of model, the colon cancer model, has reversed back to the normal weight without tumor inoculation, suggesting lowered tumor burden in these mice. We also assessed the T-cell activation. So we looked at the CD8 cells that are activated, so interferon positive, and in this case, we looked at the cells in the spleen, and you can see that there is a higher percentage of activated T cells in the spleen, and also more CD4, CD8 T cells in the combinatory treatment per tumor mass, and that suggests we succeeded in activating the immune system as a result of successful delivery. So how about giving up the drug altogether and just rely on a cocktail of nucleic acid combination to treat? So we are basically relying on a treatment, and if we go with what we call personalized medicine, you may know that not all patients, for example, are suitable for immune checkpoint inhibition in cancer, and they have to personalize and stratify the patient. So we want to find a treatment that could be suitable for the patient depending on their immune checkpoint profile. So I'll explain some of the terminologies here. You may be aware of the Nobel Prize awarded for the discovery of PD-L1 and for CTLA for as immune checkpoints inhibitors for treatment of cancer. So what do we mean by immune checkpoints? So cancer cells and immune cells express certain molecules. Some of them are inhibitory, and some of them are stimulatory. So for 1-PPL, for example, or OX40 ligand or its receptor that could be expressed on the cancer or the antigen-presenting cells, if they're activated, they could have a stimulatory effect. But there are other pairs that have immune suppression activity. So what we want to do is, in case of the immune suppression activities, such as PD-L1 and CD47, we would like to inhibit this. So we would give an siRNA, but the stimulatory one, we want to boost them by giving an mRNA that increase the expression of the receptor or its ligand. So to prove this concept, we relied on combination of, in this case, OX40 ligand that is expressed on the cancer cell and the antigen-presenting cell. And we wanted to silence PD-L1 on the same cell. So how can we do that? You can use lipid nanoparticles to co-encapsulate the siRNA and the mRNA. And as David has highlighted, this will be taken up by endocytosis or various mechanisms inside the cells. There will be endosomal escape, but the site of action is different. So for siRNA, you really want them to reach the cytosol. So is mRNA, but if you're dealing with a plasmid, which you can also be used, you need it to go to the nucleus. And this is where delivery becomes very important. You don't want only to deliver to the cell, but it's important to deliver it to the right organelle in the cell. That could be mitochondria, cytosol, nucleus, etc. So this is how precise we need to be. Okay, so we published this paper last year, and we have shown how we can use nanoparticles to reprogram the immune checkpoint interactions for cancer immunotherapy. So this is our hypothesis here. You have the PD-1, PD-L1 expression, which you want to basically silence using the siRNA, and you have the OX40 that is preferentially bound to the X40 ligand on the immune cell. So what happens if we are boosting this interaction and inhibiting the PD-L1, PD-1 interaction using the SNALP transfection? So the first thing is you want to make these lipid nanoparticles as small as possible. You want them to co-encapsulate the nucleic acid to the same degree. So for example, it's very easy to encapsulate siRNA that's very small in size, but leave the mRNA outside the particle. So we had to ensure that you have equal ratios as when you introduce it to the particles. So these are the components that we use. I'm not sure if the pointer is working fine, but I'll just verbally explain it. So in this case, we've used what we call MC3 ionizable lipid, which has been used in clinically approved formulations. Cholesterol is required to give flexibility to these particles. The neutral lipid is helping the fusion with the endosome, and the ceramide PEG is to protect these lipid nanoparticles from the macrophages so they don't get cleared very quickly after you administer them. This is how the particles will look like. You can see that they are about 100 nanometers in size. You can see the concentric layer of the lipid with the nucleic acid, where we believe it's kind of sandwiched in between the layers, and you can either form them with a siRNA or mRNA or the combination, and the shape does not change. It's very important you measure their size and homogeneity. So their size is below 150 nanometers. It's hard to get them to 100, but anything below 200 is still acceptable. The body dispersity, meaning how different the size is if you're doing nanoparticles, anything below 0.25 is acceptable. And as I mentioned, they are neutral in physiological pH. And this is very important because if you administer cationic particles, they induce all sorts of complement activations, which is obviously not desired. So the low pH makes them charged, so that allows the complexation, but also it allows the escape from the endosome later on after internalization. And you can see we have high loading of the nucleic acid inside the particle. At this stage, we didn't know which nucleic acid is being loaded. So we had to find out the composition. So what we've done here is we degraded anything that's non-encapsulated using RNAs. Then we retrieve those nucleic acid with binding to heparin as an anion, and then we run them on the gel. And we compare the intensity of the mixture below basically below encapsulation, looking at the ratios, but also the ratio in the dissociated SNAPs, stable nucleic acid nanoparticles. And because the ratio was maintained, we believe that we have kept that ratio inside the particles. And this is important when you're dosing patients, because you want to make sure that the dose you claim is the dose that's being given. So we then did transfection studies. So transfection studies means you take the cells, you expose them to the particles, and then you measure the expression of the PD-L1 or OX40 on these cells. So the y-axis is the PD-L1, x-axis is OX40, and what you need to see is reduction in the PD-L1 expression, either using the mono particles or the combinations, which you kind of see. Maybe I'll use this pointer. No. And then with the OX40, you're looking at the shift from the left to the right. So in the untransfected, you don't see any OX40 ligand expression, but with the OX40 particles, you see that shift to the right. So we have successfully transfected the cancer cells with both OX40 ligand and SIPDL1. And this is just measuring your level of transfection. For example, the PD-L1 expression is reduced in the mono or combination therapy, and the OX40 is also increased, and you don't interfere with the expression of one target over another. So if you have two targets, you're successfully transfecting the cell with both. The toxicity of these particles were only high at high concentration. So this is really our working concentration here, where the cells are within the above 80% cell viability. And because we are hypothesizing that the macrophages will play a role, we transfected immune cells, and in this case we used J774 as a model, where interestingly we found, in addition to expression of the PD-L1, we have in the negative particles, where you don't have any specific PD-L1, you have upregulation of PD-L1 receptor, which means that your macrophages are also being activated in addition to silencing. And that's confirmed by expression of CD80 and CD86 in macrophages that have been transfected either with the OX40 ligand SIPDL1 or the negative sequence, which is our sham control. So then, does it really transfect the tumor? So these are mice that were administered intratumorally. So this is the tumor site. We label the particles, so it's in yellow, red, and this is the luciferase, our model target. And what you can see, if you're administering these particles intratumorally, they stay in the tumor, they transfect the tumor, and they express the luciferase. If you give them intravenously, they mostly transfect the liver. And this is how the ex vivo images look like. So you have specific effect in the tumor with the intratumoral injection, but with the intravenous, they go to the liver and the spleen. So because this is a proof of concept to show if combination therapy works, we went with the intratumoral injection for therapy. So these are just bar charts showing quantification of the ex vivo images, so I'll ignore them. But then I want to show you the distribution of the particles in the subcellular components. So these are tumor lymph nodes, tumor draining lymph nodes, and these are tumor cells. And we want you to know whether these particles are in the B cells, T cells, or dendritic cells. And you can see here that all the immune cells are actually taking up these particles. In case of the tumor, you have the CD40, you have the myeloid and non-myeloid cells. So the tumor cells are CD45 negative cells, and the immune cells are the CD45 positive cells. And again, you have distribution in both cells. But if you look at the MFI, which tells you the quantity of the particles taking up in these cells, you can see that, for example, the dendritic cells are very hungry to these particles, while in case of the tumor, also the immune cells are preferring these particles over the cancer cells. So the good news is we're able to target both cancer and non-cancer cells to exert the effect. So what happens if you look at the tumor growth here? You can see that the tumor growth is reduced when you have the combination therapy. You also see that in case of the immune cells in the tumor draining lymph node, you have a higher number of CD4, CD8 cells. Also the activated T cells are very clear. If you look at the tumor here, you don't see significant increase in the CD4, CD8. So what probably is happening, you're injecting them, they're retained for a bit of time, but then they go to the tumor draining lymph node and exert their effect. So we sent this to publication, and the reviewers wanted to see survival results. So we had then to break the different groups. So we have the individual treatments included this time. And what we can see is only in the combination treatment, you have approximately 40% of the treated mice cleared that tumor. So the tumor completely disappeared. So we re-challenged these mice, and we waited for the tumor to disappear, reintroduced the tumor again, and these mice did not develop the tumor anymore, which means that they have been vaccinated for the tumor. But if you use comparable age mice, they grow the tumor with delay because they're older. So we really concluded that we're able to vaccinate the tumor by using this type of combination treatment. So what we reflected back on our original hypothesis, and what we think was really happening is that you're giving these lipid nanoparticles inside the tumor. You are transfecting the cancer cells and immune cells, and you are reprogramming that interaction crosstalk between the tumor cells and the macrophages and the T cells. We also believe some of these particles drain into the tumor draining lymph nodes and exert a distal function, which probably was responsible for that protection against tumor recurrence. I would like to thank the team who have contributed directly or indirectly to the generation of this data over the past 10 years, all the PhD students and postdocs, also my collaborators on the various projects, and the funders, and you for listening. Thank you very much.
Thank you, Khuloud. That was a wonderful talk and a lot of information. I liked how you started with bisphosphonates, which is what, thank you. One thing I know something about. But I had a question. I mean, this is very impressive. Do you know if this is already moving forward? Is it ready to go to patients or larger animals?
Okay, so I think it goes back to our problem with science, IP, industry, and so on. So, for example, we submitted this for patent, and the answer we got, although you're combining PD-L1 with SICD47 using non-antibody approach, people have tried combination of antibodies, and that's why this is not going to be inventive, though it's novel, right? So I guess maybe it's the way we probably have to see how the landscape of exciting science and therapeutics versus commercial interest should move forward. But so what we're trying to do now is look at some angles that probably would make the next step slightly more inventive. So we realize that it's not just about doing science, but clever science. So.
Yeah, but you think scientifically it's ready? You understand it sufficiently?
Yeah, absolutely.
Yeah. Do you have a question? Yes, somebody's the audience makes his way
Must see
to-
us. I cannot see anyone because the light is here, so apologies.
Microphone
Yeah.
1 first, and then microphone 2, yes. So.
Thank you, Professor Khuloud. Fascinating talk, especially towards the end, that when you know that your mice is immunized against cancer, that brings a lot of hope into the future. I've got 2 questions. What are your thoughts about extending your treatment into metastatic disease, i.e. targeting the primary tumor, and would the immune system attack the rest? And what are your thoughts about immunizing high-risk phenotypes, say like BRCA gene, like breast cancer? Would you vaccinate your mice against breast cancer? Thank you.
Mm-hmm. I think this strategy would be applied to any type of solid tumors where you can access the tumor, for example, whether breast cancer, brain cancer. So we currently have a project on brain cancer to apply the concept to treat brain cancer. So there is a scope. It's about really the funding. If we can have funding to support something into BRCA therapy, but the rationale is very strong. The first question was about, I guess it's more of a technical, so when you re-challenge the tumor, the home office tells you you have to re-inject it, the same type of tumor. So, but logically, I would imagine we could do intravenous injection and look at the protection in the lung. And I don't think it should matter because clearly the immune response, when we inject the second tumor, it's on the other flank. So it's an interesting idea, especially with the B16 model, because you can get it subcutaneous or a metastatic lung. That's a good point.
Microphone number 2.
Hi. Lutz Willer, Hoffmann La Roche in Basel. I wonder about the CD47 expression in other tissue than the tumor. So if you intravenously inject, whether you have some potential toxicity to other organs expressing CD47 under these circumstances.
There are monoclonal antibodies for CD47 that are being given. It is there, and probably that's why there are toxicity with the monoclonal antibodies. What we've seen here is clearly no changes in body toxicity, but that's probably to do with the fact that particles passively target tumor cells. So they wouldn't, for example, extravasate everywhere. They will be going to the liver. So what we have to worry about is liver toxicity and the tumor. So from the data, maybe targeting CD47 in the liver wasn't a big problem.
All right. Thank you, Khuloud.
Thank you very much.
Thank you, everyone. So now we move on to across the pond. We go to Harvard in Massachusetts. To John Mannis, I hope. Hi, John. How are you?
Hi there. Thank you. Good.
Hi. Right. Let me introduce you. So, Dr. John Mannis completed his undergraduate and medical studies at the Johns Hopkins University and the State University of New York in Syracuse, with subsequent training in internal medicine and fellowships in hematology, oncology, and transfusion medicine. He then moved to Boston Children's to pursue a postdoc with Dr. Frederick Alt, where he worked on basic mechanisms that control programmed DNA breaks in B cells of the immune system. In 2004, he joined the Department of Lab Medicine at Boston Children's, running a lab that's working to understand basic mechanisms that control B cell movements. Errors in the repair of programmed immune system breaks lead to immunodeficiency, and his lab generates and studies various mouse models of human immunodeficiency. Since 2010, he's the director of the Therapeutic Apheresis Unit and the Stem Cell Collection Facility at Boston Children's, and since 2022, the chief of the transfusion service. He participates in over a dozen molecular therapy trials, including trials to improve mobilization and collection of stem cells in sickle cell disease. He's part of the Cure Sickle Cell Initiative launched by NIH, where he chairs the Committee for Apheresis Research and oversees a clinical trial to collect stem cells for a national repository. He's a co-investigator in multiple clinical trials testing gene therapy approaches to ameliorate sickle cell anemia. So he's the guy to talk to about how to cure sickle cell disease. He also is a native New Yorker, and so he knows what a bagel and a schmear means. I saw that you invented something called a schmear RNA, so I hope you can explain to us what that all means. Thank you, John.
Thank you, Serge, for that generous introduction. And thank you to the organizers for this. Let me share my screen and mute myself. Sorry. So what I want to, what I'd like to bring up today is some therapeutics that we're doing to understand and treat sickle cell disease. And this is a collaborative effort, and in no way can I take credit for this. This trial that I'm going to present today was a single institution trial that's now turned into a multi-institution trial that is really trying to target problems in sickle cell disease, which I'll bring up, undertaken by the sponsor, David Williams, and Erica Esrick, the PI here, and I'm part of that team that's doing it. So sickle cell disease is a disease that has multiple manifestations, but really comes about because of a single point mutation in the beta globin, in the hemoglobin protein. The beta subunit is abnormal, and the pairing of globin structures induces hemoglobin that has different oxygen tension characteristics. Its capacity to carry oxygen is greatly reduced, but moreover, what happens is there's an imbalance of these proteins in the cell, and there's an abnormal deformation of the cell into a sickle shape. That's the characteristic that's seen pathologically on smears and in cells, but importantly, what this brings up is that these have long-term end organ damage. So there's a chronic hemolytic anemia because the half-life of these cells is shorter. There's acute and chronic problems. There's vascular end organ damage due to ischemia, stroke, lung hypertension. These are all devastating side effects. And with transfusion therapy, we're able to extend the lifespan of patients into their 40s, 50s, and 60s in the present day. Nevertheless, these are devastating side effects for children, especially for children which suffer from stroke and from lung injury. That's a very common problem. There are multiple approaches to this genetic effect. It's really dozens and dozens of therapies that are now being developed, genetic therapies. In terms of medicinal therapies, it's quite amazing that from the early 90s until only 5 years ago, no drugs were approved in the United States for sickle cell anemia, for sickle cell disease. And the first drugs, really, the disease-modifying drugs, came out about 5 years ago. The genetic therapies have been in production for many years. And there are many approaches. There's a gene correction, and that's in development, actually using CRISPR-Cas mechanisms to go in and correct genes without inducing double-strand breaks. There's the induction of double-strand breaks, and that's actually in trials now, where the enhancer for this protein BC11A sits on the globin locus. That's here. I don't think my pointer is working. But there's the induction of hemoglobin F. What's been approved for thalassemia that may also work for sickle cell disease is gene addition. Obviously, we have a defective beta globin gene. Why not just put an extra wild type gene in, a normal healthy gene? And that is actually been approved for thalassemia and is in trials for sickle cell, both in Europe and in the United States, approved in the United States for thalassemia. So what is fetal hemoglobin? Why the focus on fetal hemoglobin to treat sickle cell disease? Well, fetal hemoglobin is what, as it's called, is what fetuses use to preferentially take oxygen from the mother so that you could actually oxygenate a fetus. And what we see is in the prenatal age until birth, we have gamma, which is fetal hemoglobin, being produced. At birth, there's a switch. The switch occurs that we make beta globin, and that replaces gamma globin. This switch is very, very important because what we see is when neonates with sickle cell disease do not have any symptoms. They're healthy. They're fine. So there was thought that there was a protective effect of fetal hemoglobin in fetuses. So what was next understood, and this was done over 60 years ago, is that sickling, again, was not commonly seen in newborns. And this was a very, very early identification, despite the cells weren't sickling, despite mother cells sickling. So this really led to an understanding that there was a protective effect. In the 90s, it took another 30 years after that to really go into the clinical studies. If you look at symptoms with high fetal hemoglobin, you can see that if you have the higher the fetal hemoglobin you have fewer pain incidents, acute chest, and actually there's a protective effect for death. So what we do know is that the higher the fetal hemoglobin, the more protective it is. And that's something so that became a potential target to treat this disease. Now, what's different with fetal hemoglobin? If you look at this oxygen saturation curve, what you see is that hemoglobin SS, which is sickle, really has less oxygen saturation than all the way at the bottom. What you see is hereditary persistence of fetal hemoglobin. These are patients that are born with high fetal hemoglobin levels. And what we see there is that they're able to saturate their hemoglobin with oxygen at a much, much more effective level than even wild type and even normal hemoglobin. And these patients with fetal hemoglobin, even in the presence, hereditary persistence of fetal hemoglobin, even in the presence of sickle, have a protective effect. So there was a search for the molecular targets of fetal to adult globin switch. And if you look at the fetal hemoglobin, at the globin, beta globin locus, you can see that there's beta globin, there's a pseudogene, there's fetal hemoglobin. So what are the regulators of this fetal hemoglobin? And there was a genome-wide, a GWAS analysis done, and of course looking at patients with hereditary persistence of fetal hemoglobin. BCL11A was found as a target. Now, BCL11A does many things. It's found in B-cell lymphoma. That's why it's BCL11A. It was actually the 11th cloning factor found. So it has many functions, including actually regulating myelopoiesis. So you could knock it out in mice, as we tend to run to mice for these experimental. And if you knock out mice, BCL11A in mice, even in mice that have sickle cell disease, you can see that there's a reversal of this phenomenon. But you can't knock out BCL11A in all tissues because you would not have myeloid cells and nor any B cells. So you really want to, if if you want to deplete BCL11A, you need to do this in a tissue-specific fashion, and you need to do it specifically in erythroid cells. So we need to flip the switch. If we think BCL11A is interacting with this locus control region that can cause the switch from gamma to beta, then we need to knock it only in erythroid cells after birth. So the goal was to try to take this therapeutic into the clinic and really knock down BCL11A using RNAi technology and lentiviral vectors. Now, lentiviral vectors have been around for 20, 30 years. That really is bringing the delivery into a cell. The difference here is that we want to bring this into a cell long-term, and not only that, but bring it in to hematopoietic stem cells. And this will include, of course, B lymphocytes, but not turn it on in all hematopoietic cells, but only turn it on in the erythroid lineage. So we developed a vector. This was really developed, and this is really 20 years of work, understanding locus control region, understanding which promoters go on, and how strong these promoters are. And this really has something of importance, because delivering this vector and a strong promoter is quite important in this day and age, because this vector has been used for other diseases like adrenoleukodystrophy with notable side effects that I could answer in questions or in emails afterwards. So we engineered an schmear, as we put an shRNA, and this is mimicking a microRNA, which will be expressed specifically with regulatory elements for the beta globin locus and with a bovine growth hormone, really with a strongish promoter, but not a super strong promoter, and without the regulatory elements of the LTR of the virus. So what we could see here, and I'll be brief and to the point with these, is that if you take this vector and you transfect it, and this is into CD34, into stem cells, whole blood stem cells, you could see that there's an induction of gamma globin here on the right. Apologies again if the pointer's not working, but we could see that there's an induction of a depletion of the protein and an induction of fetal hemoglobin. The Western on the left, the protein, the end product, the deregulation. If you take this into mice, for example, and take stem cells into mice that have sickle cell disease, this is the Berkeley model, and on the left, it's empty vector. In the middle, what we could see is CD71 represents young cells, and in sickle cell disease, because there's such a high turnover of cells, there's a lot of hemolysis, there's a lot of CD71 expressing young cells being produced. You can see in wild type it's only 2%, but in sickle it's close to 40%. There's an amelioration of the phenotype if you include this shRNA. So we were quite excited to understand that there would be some therapeutic benefit to this with an improvement of the sickle cell disease. Not only is there a protective effect by making more fetal hemoglobin, there's a decrease on the right side here. We could see the hemoglobin S protein. The ratio and the production of hemoglobin S goes down. So there's a twofold effect of putting in the schmear RNA. You don't allow BCL11A to regulate, to cause the switch to the wild type, allowing it to make fetal hemoglobin, but you also decrease the amount of defective beta sickle that you're making. So what did we do in the clinical trial? This was a single center pilot and feasibility study. We had 10 patients in total, descending age, and we brought it down to very young. We were able to go down to 6 and 7 year olds, not lower than that. Really, to do this, we know that transplantation helps. You had to not have a matched sibling donor. So you had no other option. You had to have a severe clinical phenotype, that you need chronic transfusion therapy, that you did have acute chest or vaso-occlusive disease. So you had side effects of sickle cell. And we had one patient with recurrent priapism, a side effect, of course, a manifestation of vascular disease. So what I worked on, and I want to take credit just for that part, because there's a whole big team here, was trying to collect the stem cells and understanding how we collect stem cells, and that's done with plerixafor. That's a whole separate discussion and study in itself. But then what happened is you collect the stem cells, manipulate them in vitro, put the virus, allow them to grow, and then have the infusion of these gene-modified cells. And the study had a 2-year follow-up. And actually, we're out 3 years now, looking at how much fetal hemoglobin we're making. And really, the endpoint was sickle symptoms. So if you look at the patient population, the inclusion criterion, what we have here is stroke. I want to point this out because stroke is not a reason in the newer trials of others to be entered into the trial. Nevertheless, we included stroke because this was very early on. And we had many patients were on chronic transfusion regimens, others weren't. So really before infusion, we didn't really have adverse events. Really, the adverse events were related to central venous access devices. In other words, to take out stem cells, to do transfusion, there was a vascular access device, a catheter. And this is what caused the majority of infections and clots and problems. Many patients had vaso-occlusive pain coming in. There was acute chest. There were no neurological events. Priapism was a symptom in one of the patients. Was there amelioration of the clinical phenotype? There was. And what's very, very striking here is that really going in, there was anemia. And at the latest time point, we had an increase, and I'll show this to you, of hemoglobin. So we were able to raise the hemoglobin of the patients. There were no respiratory neurologic events. And actually, there were no additional transfusions. These were patients before their transplant, before their gene therapy, were getting transfusions. In one patient, we continued transfusions. And this is an important point because this patient had severe neurologic disease. He agreed to go along with the gene therapy, and we were hoping to understand whether we can substitute gene therapy with transfusion. We're hoping to do this in the future. We don't know if it's safe right now to substitute, to let go of his transfusion therapy. Again, this is subject number 3. No one else is being transfused, and there have been no severe clinical events, except for one patient. We did have one patient who did have pain for the first year after gene therapy, a little bit under a year, and that pain has now, at 2.5 years, subsided. If we look at time since infusion, and for the sake of time, I'll go through these slides relatively quickly. There's a long-term effect. If you think of these, this is months on the x-axis, even up to 3 years, and these have come out. We have hemoglobin, fetal hemoglobin induction. So there's long-term induction. Remember, this is a virus introduced into the stem cells, and what this is doing is expressing this microRNA, the Schmier RNA, and we could see fetal hemoglobin production. If you look at the number of F cells, and what this means is we do flow cytometry, and look at the percentage of cells that have fetal hemoglobin, you could see that 60, 70, 80% of red cells in these patients with gene therapy, here is the pre-gene therapy all the way on the bottom here, number of cells, we have a very, very robust number of fetal hemoglobin inducing cells. So there's long-term and substantial number of cells with fetal hemoglobin. A busy slide here. We did have, apologies, one failure, BC1110, the fourth from the bottom. And in this patient, for unknown reasons, did not get a benefit. If we look at vector copy number, and what that means is if we look at the number of copies of what we introduced in the stem cells in the peripheral blood, actually this is in blood cells, not stem cells, we don't see that there was long-term selection for that, never, despite putting in an adequate number of cells. This did not take. So there was a failure. So if we look at 2 or 3 more slides just showing hydroxyurea treatment has been the mainstay of therapy inducing fetal hemoglobin for the past 30 years, introduced in the early 90s. We could see that on the right, what we have is how much fetal hemoglobin we induce with a drug. We could see a much, much higher percentage of over the threshold being 20% of induction. So a much more, a higher number of cells that have fetal hemoglobin in patients with gene therapy. So we think that even though you can induce fetal hemoglobin with hydroxyurea, you may not be doing it. Now, one of the biggest problems, and this is one of the final slides I have, is how do we judge success? In other words, we could look at protein or, but looking only at side effects and organ damage rather, vaso-occlusive disease is only one measure. Could we do anything in vitro? And collaborators, this is John Higgins, but also collaborators in Paris, Pablo Bartolucci, doing these studies, looking at polymer formation in cells. And what we have is really a tube with oxygen, changing the oxygen tension. And what we see is that the corrected cells really have, hopefully the pointers are working, an oxygen saturation that is improved after gene therapy. So we do think that we could use this tool as a surrogate marker to know right away. And if we look at time, and this is after infusion, 2 years after infusion, we could see that the red cells containing a minimum of 10 picograms of hemoglobin F, we could see that there's an improvement, not only in the number of cells that are getting fetal hemoglobin that are protective using this in vitro assay. So really, we've had these 10 subjects come in enrolled, including children. There's a GMP product that was developed specially for each of the patients using their autologous cells. There was extremely efficient transduction of HSCs, no adverse events related to vector. This is important because this is a very hot topic now. If putting a vector in and putting a virus into stem cells, does this have side effects? We have not seen this in our studies. Expression is lineage specific. Remember, not only are we putting the smear in the microRNA, expression is limited to the erythroid lineage. We have broadly distributing hemoglobin F with a high percentage per cell of hemoglobin F and a significant attenuation of side effects. So we do think BC11A is an effective molecular target for sickle cell, whether we have new drug therapies to put gene therapy out of business. I hope so. I'm looking for colleagues in the audience in this meeting to put us out of business because we don't want to be doing this in everyone if we don't have to. But this is a very, very important axis to go in. We do have a phase 2 study going through. This is in the States for fetal hemoglobin with 4 centers. And again, new targeting methods and viral vectors, really with the Gates Foundation and other institutions, and looking at immune conjugates. So the phase 2 study has been launched actually September, sorry about that. and done by Mark Walters in California and David Williams here. So really, that phase 2 is similar to the phase 1 study I've just presented, except multi-institution. Many people, really Dave Williams is the sponsor, has really pioneered this for 20 years working on gene therapy. Great collaborator, Erica Ezrick, who's the PI here. Many other, I can't stress cell manufacturing, the NIH, trying to get the end studies, John Higgins, Pablo Bertolucci, Nicolas Hebert, others and other institutions. And I really have a lot of people to thank for this. Thank you, and I'm happy to take questions.
Thank you, John. I think there's questions in the audience. I have a lot of questions myself, but I think I'll start with the questions from the audience. So, microphone number 2.
Thank you very much for your talk. I just had a question about kind of a forward planning with this, and obviously using hemoglobin F, there's a consideration around pregnancy and whether any of these female patients going forward. And I was just interested in how that's been incorporated.
Absolutely, it's a concern. We don't know if raising hemoglobin F to the level that we have done will have effects of pregnancy. Patients with a persistence of fetal hemoglobin have had successful pregnancies. So it's really unknown, but it is a concern. All these patients did go through fertility preservation, and it is a known side. It's a concern of ours. I am glad you brought this up.
I very much enjoyed your talk. It does sound like finally there's something that might work for these poor patients. One of your flow cytometry graphs that you showed that there was the levels, the number of cells that were expressing the fetal hemoglobin. And the range, I think, I'm sorry, I might not have seen it correctly, was somewhere between 40 to 80%. And I know that I suppose this is a relatively small study because this is how you start. What was the reason for that variability? Was it the demographic of the patient, or was it something related to the actual intervention?
It may be the actual intervention. There's a slight signal. It's too small a study, and it's too few, but there's a slight signal for vector copy number. In other words, we may not be introducing enough of it, of the schmear into the cells. And the second part is that there is still endogenous production of non-corrected cells. So there's this balance of the longevity of the cells, of what is corrected. So there is a variability, a cell-to-cell variability, and we're looking at that by looking at single cells.
And there are some experimental models, aren't there? There are animal models that can be used in testing these kind of interventions in animals. Has that been done? And was the same kind of... Because of course, you have uniformity of the patient, don't you, in a sense? But does the same variability exist in a relatively homogeneous population?
Not as much. You're absolutely correct. It's a very good point. In the mouse models, it's much tighter. But in patients, it isn't. So we don't know if it's, there are additional factors. One of them being, and I should bring that up, is are there other alpha gene defects, for example? Looking at alpha thal, deletion of other genes. So there are non-BC11A, in addition to the amount of reduction of BC11A we have in the erythroid, there are other factors that have to do with the proportion of the beta globin and alpha globin genes. And what that contributes to is the half-life of the cell.
Okay, thank you.
My question, John, is well, a few, but let me restrict myself to just one. How do you see this therapy in relation to the other therapies that are all out there, the CRISPR-Cas, the hydroxyurea, and so on? Could you tell us a bit about it?
It's a very good question. It's a dizzying landscape right now. I'm in Boston. Behind me, a mile away, there's about a dozen biotech firms, and as well as other laboratories, please, of course, doing all sorts of approaches. There's the addition of the gene, the second slide I pointed out. There's CRISPR-Cas. Why knock out the protein when you could simply delete the DNA of where this protein sits on the beta globin locus? So why introduce a virus when you could just run in, alter the DNA, and run out with introducing the machinery, the protein machinery? So it's extremely, extremely exciting, dizzying. It's a David and Goliath. I'm bringing in smear RNAs, which are nostalgic to some students. But what I am bringing up is that we know this works. We have 4 years of experience. We haven't seen side effects. Putting these vectors in with other promoters, we do know can cause leukemia. So there is that concern, but we have not seen this coming out. And what we're worried about is that these effects may be, these potential leukemic effects, may be in all patients with sickle because of the long-term damage to the bone marrow. So we'd like these therapies earlier and earlier. So I do think there's an importance, two things. One is, let's try all genetic therapies, but of course, as I brought up, I'd love drug interventions. Two is really understand the importance of treating this disease early on in childhood, and also the effects it has on stem cells, and trying to understand those effects.
Thank you, and I assume it's also cost may be an issue for some of these other, and maybe this one too.
I'm in my ivory tower here, so we never talk about money. But I will say that for thalassemia, that was just approved last month in the United States, a similar treatment dropped my jaw that the guess is $2.8 million per dose. That is not a small undertaking. That is not, there's a lot there. There's a lot to unpack. Now, if this is indeed a one-time cost for a lifetime, sure, of course. But I will say that it hasn't been approved by the EMA. I'm sure the UK is kicking cans or whatever they could do about this, and rightfully so, because we'll have to select there. Do we take patients where there are no other drug interventions? And, preventing a stroke in a child is a big deal for long term. So I'm not trivializing the cost. I have nothing to do, well, I don't know what I have to do with it. But this is the reality today.
Thanks. Thank you, John. I have many more questions, but I'll ask them some other time. I think we've come to a conclusion. Thank you, Ellen. Thank you, John. So we've come to the conclusion of this session. I hope you enjoyed it. I did. Amrita?
I enjoyed it very much indeed. I think it's fantastic, the approach of the RNA-based medicines. Fantastic.
So thank you for coming, and thanks to all the speakers and for the organization of this symposium. Thank you. Bye.